So safety is the big concern. And, a lot of this, people feel like corners have been cut. So how do we get from a normal 10-year process down to a 1-year process? Do you want to just summarize some of the stuff that changed?
Yep. I think that's probably the most important questions to answer.
Mm-hmm.
Because when something that on the average takes 5 to 10 years, how do we manage to do it safely and properly, say, in 7 to 8 months?
Mm-hmm.
So I'm going to answer this in several parts. Number one is we happen to got very lucky with this particular coronavirus, which means this turned out to be a virus. It was not difficult to develop a vaccine against the virus,
Right?
Which cannot. We have dozens and dozens of vaccines out there. Some are easier to develop, some are harder to develop. I give you the most extreme example. We've been working on HIV vaccine for the last 30 years. We're not even coming close to it because that virus is changing all the time. It's a real moving target. We just cannot find the part of the virus we can say we can develop some kind of a target against that. So there will be something we tried 30 years, we couldn't get the shoot on the moving target. It just so happened the coronavirus, it's a very stable virus. It does mutate, it does change. But from the beginning, we saw all those pictures on TV screens, on newspaper, of those spikes, those funny looking spikes. Well, from the beginning, everybody who was doing the vaccine development, doesn't matter which technology, which methodology used, everybody was going after that spike, the spike protein. Well, we got lucky. Turned out that spike protein is very stable. And if you can properly develop the vaccine or the antibody against that spike protein, you can disable this particular virus. So the good news number one is this turned out to be one of the easiest vaccine to develop.